A very good afternoon to all the NEET aspirants. This is your Prasanna ma'am from NEET World. Today we are going to discuss on the high weightage topic, that is photosynthesis in higher plants, and we are going to cover all the light reactions including non-cyclic photophosphorylation and chemiosmotic hypothesis. So yes, let us get started. Firstly, we have to know what is the standard equation of photosynthesis and what are the synonyms for photosynthesis. Photosynthesis is also called as an anabolic process or oxidoreductive process, anabolic or oxidoreductive process, or it is also called as physicochemical process. It is also called as what ma? Physicochemical process. This is called as anabolic process because it uses simpler inorganic molecules like carbon dioxide, water and sunlight. In the presence of chlorophyll, it builds up the complex organic molecule like glucose. That is why this photosynthesis is said to be an anabolic process. It is said to be oxidoreductive process. This is because first you understand the standard equation of photosynthesis, which is 6CO2 plus 12H2O in the presence of sunlight and chlorophyll, which builds up C6H12O6 plus 6H2O plus 6O2, which is liberated out as byproduct. So yes, here in this equation if you look into, carbon dioxide from the reactant side is getting reduced, carbon dioxide is reduced to glucose, and water molecule is oxidized to oxygen. So if you look into here, both reduction and oxidation reactions are taking place simultaneously in the photosynthesis process. So it is also said to be as oxidoreductive process. This is said to be physicochemical process because the physical energy, that is light energy in the form of photons, light energy is taken in the form of photons, and this is converted to chemical energy, that is ATP and NADPH. As light energy is converted to chemical energy, this is also called as physicochemical process. So yes, photosynthesis is also called as anabolic process, oxidoreductive process and physicochemical process. So if you look into the standard equation of photosynthesis, we are clearly knowing that this is a process by using inorganic molecules like carbon dioxide, water in the presence of sunlight, that is photons. It is building up complex molecule, that is complex organic molecule, that is glucose, and water, oxygen is liberated out as byproduct in the photosynthesis. This is identified by many sequential experiments done by many scientists. So we have to have the clear idea about early experiments. Many a times the questions are asked beta. So very first experiment that we have to look into is variegated leaf experiment. Wherein the plant which is of leaves which is of many pigments, many colors, like it will be pale yellow, pale green and dark green, such type of leaves are taken, or one more plant which is totally green and it is covered by some black stripe or paper. A part of leaf is covered by black stripe or paper. This plant, when it is exposed to sunlight, for some time, and when this leaf is taken out for the starch test, what happens ma? The leaf which is covered by this black stripe, okay, leaf which is covered over it by black stripe, this leaf will after conducting starch test, the leaf part whichever is exposed to sunlight, the leaf part whichever is exposed to sunlight, forming starch iodine complex, this turns to be blue black color. This turns to be what ma? Blue black color. And the one which is covered by the black stripe cannot take the sunlight, thereby it cannot perform photosynthesis and form starch. This turn out to be pale color. Okay, this is the initial experiment which is done to say that starch is formed at the end of photosynthesis, starch is formed as the end product, and photosynthesis takes place in the presence of sunlight. And the next is moles half-leaf experiment, that is moles half-leaf experiment. This experiment is to prove that carbon dioxide is essential for the process of photosynthesis. This experiment is to prove what ma? Carbon dioxide is essential for the process of photosynthesis, wherein plant which is exposed to sunlight, a part of the leaf is kept into a beaker containing solution. It is kept into a beaker containing solution KOH. Half of the leaf is immersed into the beaker. Okay, half of the leaf is immersed into the beaker which is containing KOH solution. So the KOH which is present in this beaker will react with carbon dioxide. It will react with carbon dioxide, forming a potassium carbonate, and water. So yes, it will completely utilize carbon dioxide. Complete carbon dioxide which is present in this half leaf is utilized by the KOH. It is reacted with KOH. Now there is no carbon dioxide in this half leaf which is immersed in the beaker. The next half leaf which is exposed to sunlight has carbon dioxide and it will conduct the photosynthesis. So this leaf, when it is conducted with starch test, half of the leaf which is exposed to sunlight turns out to be blue black color, and half of the leaf which is kept in the KOH solution will turn out to be pale color, indicating the absence of photosynthesis. Indicating what ma? The absence of photosynthesis. Okay, this is moles half leaf experiment conducted by mole. Okay, and the chemical used is KOH, where half leaf will turn out to be blue black and the next half is pale color. Next, if you look into bell jar experiment done by Joseph Priestley. Okay, what is it ma? Bell jar experiment done by Joseph Priestley. So here if you take in the bell jar, firstly a lightened candle and a live mouse is kept and closed with a bell jar where no air can enter from outside to inside. After certain period of time, the rat has dead and the candle also has put off. The same experiment is repeated by having a mint plant inside the bell jar. If he checks the bell jar after some period of time, mint plant, rat and candle, everything is fine. So with this he said that the gaseous exchange happened between mint plant and rat and candle. Okay, there is a gaseous exchange between the plant and animal candle. Okay, so yes, what is that gas? He didn't identify it. Bell jar experiment is done in 1770, but then oxygen is discovered by Joseph Priestley in 1774. Okay, in the year 1774 oxygen is discovered. Initially he said that a gas which is exhaled by the plant is inhaled by mice and the gas which is released from the mice is taken by the plant. Later he confirmed that gas as oxygen. Okay, in the year 1774. Now comes the experiment of Jan Ingenhousz. Jan Ingenhousz, wherein he said that sunlight is essential for the process of photosynthesis. Sunlight is essential for the process of photosynthesis, and water, sunlight, water is essential, and oxygen is released at the end of photosynthesis is also said by Jan Ingenhousz. How did he done the experiment? He has taken a beaker full of water and a tistle funnel with a hydrilla plant, and this tistle funnel is inverted into the beaker and a test tube is placed over the tistle funnel. Two such setups were taken. One is kept under sunlight and other is in dark room. Okay, one is kept under sunlight and the other is in dark room. The beaker which is kept in the sunlight, after certain period of time, if he observes, it has formed few bubbles in the test tube. He can observe few bubbles in the test tube. When he has taken the test tube and kept a candle in front of it, or a light in front of it, it has burnt out so much, like it has burnt more vigorously, indicating the presence of oxygen. This indicates the presence of oxygen. Okay, what did he do, ma? The setup which is kept under sunlight, after certain period of time, it has shown the water bubbles in the test tube. When the test tube is taken and checked out for the presence of oxygen, the candle which is kept in the mouth of test tube has burnt out more vigorously, indicating the presence of oxygen, and the one which is kept in dark room has not shown any response, indicating the absence of photosynthesis, and there is no oxygen formation. Photosynthesis takes place in sunlight and water is required for photosynthesis. Oxygen is evolved from the process of photosynthesis is said by Jan Ingenhousz experiment done in the plant hydrilla. The experimental plant is hydrilla, which is an aquatic plant. Yeah. Now, sunlight is essential for the photosynthesis is said by Jan Ingenhousz. Later, the concept like which spectra of sunlight is essential for photosynthesis is said by E.W. Engelmann. Is said by E.W. Engelmann, who has set out which spectra is essential for photosynthesis. What is the experiment that he has performed? He has taken out light, sunlight or normal light, and exposed it to prism. Okay, light has exposed to prism. Prism has reflected the light into VIBGYOR. Okay, when light is exposed to prism, prism reflected the light into VIBGYOR. This light which is exposed from the prism is kept on the plant, like primitive plant, that is Cladophora. Okay, what is taken? Filamentous algae. Filamentous algae, which is the primitive plant that is Cladophora. Cladophora is placed with so much of aerobic bacteria. Aerobic bacteria. So, if you look out, ma, what did he do? He has exposed the light to the prism. Prism scattered the light into VIBGYOR. This VIBGYOR light is exposed to Cladophora, which is having aerobic bacteria over it. After a certain period of time, so much of aerobic bacteria has observed in two places, indicating two spectra. Okay, aerobic bacteria has observed at two places. One is in between 400 to 500 nanometers at blue light, and the other is at red light at 680 nanometers. One is at 450 nanometers. He has observed so much of aerobic bacteria. Cladophora conducting photosynthesis released out oxygen. So, most amount of oxygen is formed at this wavelength of light. As a result, more aerobic bacteria association you can see in between 400 to 500 nanometers, and here in between 600 to 700. That is exactly at 680 nanometers and 450 nanometers. At blue light and red light, most of the photosynthesis has taken place, and thereby most of oxygen evolution happened. As a result, aerobic bacteria has formed two spectra, okay, two spectra indicating the high photosynthesis activity, indicating high photosynthesis activity and oxygen evolution. High photosynthesis and oxygen evolution. High photosynthesis and oxygen evolution. Okay, this has led to the discovery of both absorption spectra and action spectra, both action and absorption spectra. Okay, is given by E.W. Engelmann. E.W. Engelmann has proved that which wavelength of light, under which wavelength of light maximum photosynthesis can happen, maximum oxygen evolution is seen, and he has discovered, he has given out both action spectra and absorption spectra. You have to remember it, ma. Next. So yes, after knowing out action spectra and absorption spectra, this graph is given in our NCERT, which is very important. Firstly, what is action spectra and absorption spectra? Absorption spectra means different pigments. This spectra will give you the information about different pigments absorbing different wavelengths of light and conducting photosynthesis. Absorption spectra will give you information about different pigments absorbing different wavelengths of light. Action spectra will give you the information about the pigment which is conducting high amount of photosynthesis at which wavelength. Okay, pigment which is absorbing high amount of light and conducting high amount of photosynthesis is shown by action spectra. This is the difference between action and absorption spectra. In plants, if you see, in plants there are many different pigments present. In plants different pigments are present like chlorophyll A, chlorophyll B, carotenoids and xanthophylls. Okay, chlorophyll A is bright or blue-green in color. Chlorophyll B is bright yellow, green color. Okay, carotenoids is yellow-orange colored, xanthophyle is yellow color. This is the color of different pigments which is analyzed through chromatographic experiments. So yes, these pigments absorbing different wavelengths of light is predicted under absorption spectra. You can see clearly from 400 to 500 nanometers, 500 to 600, 600 to 700. As such, you can find out different pigments absorbing different wavelengths of light. 400 to 500, 600, 600 to 700. So it is chlorophyll A that shows peak between 400 to 500 and 600 to 700 nanometers wavelength of light. So yes, maximum photosynthesis is conducted by chlorophyll A. If you look into chlorophyll B, which shows only one peak showing in between 400 to 500 nanometers. Carotenoids also the same. So two peaks are shown in absorption spectra. Two peaks are shown by which pigment, ma? Chlorophyll A. Okay, indicating that chlorophyll is conducting maximum amount of photosynthesis. Then it is action spectra. Action spectra, chlorophyll A has again shown the two peaks indicating maximum amount of photosynthesis. So there is a statement that action spectra and absorption spectra of chlorophyll A coincides with the action spectra. Chlorophyll A coincides with the absorption spectra and action spectra. This is given here, okay. Rate of photosynthesis and absorption. Rate of photosynthesis is depicted by what, ma? Action spectra. Action spectra will show you rate of photosynthesis. High rate of photosynthesis is conducted by chlorophyll A. High rate of photosynthesis is conducted by what? Chlorophyll A, both in the blue and red regions, that is in between 400 to 500 nanometers and 650 to 700 nanometers. At both the regions, absorption has taken place. So, it shows two peaks. Hence, we conclude that chlorophyll A is the pigment associated, is the chief pigment associated with photosynthesis. Clear with action spectra and absorption spectra, ma? Yes. And it is T.W. Engelmann who said that action spectra and absorption spectra, who gave us action and absorption spectra. Then it is C.B. Van Al who has conducted a milestone experiment. Who is he? C.B. Van Al, where by conducting bacterial or anoxygenic photosynthesis, anoxygenic photosynthesis, bacterial or anoxygenic photosynthesis, C.B. Van Al has said that the oxygen which is evolved in the photosynthesis is not from carbon dioxide. It is not from carbon dioxide, it is from water. Okay. By conducting bacterial or anoxygenic photosynthesis, in bacteria the electron donor is H2S. Okay. Here if you can see H2S along with carbon dioxide in the presence of light will give rise to 2 sulfur molecules and then glucose, water. So, yes, this is indicating that sulfur molecules are released out as byproducts at the end of photosynthesis in anoxygenic photosynthesis. So, from H2S, sulfur molecules are released out as byproduct. In oxygenic photosynthesis, which is conducted by higher plants, in oxygenic photosynthesis which is conducted by higher plants, oxygen is evolved from water molecule, not from carbon dioxide. Till his experiment, everyone thought that oxygen which is evolved is from carbon dioxide, not from water. He proved that oxygen which is evolved from water molecule, oxygen is evolved from water, and in an oxygenic photosynthesis H2S is the electron donor and sulfur molecules are released out from H2S. Clear beta? So, what is the electron donor for anoxygenic photosynthesis? H2S. What is the electron donor for oxygenic photosynthesis? H2O. Now, yeah. So, before going to the final reminder of your experimental discussions, okay, the questions will be asked as such. You take down this is very much important in the questionnaire point of view. Yeah. So, what is the standard equation, ma? That is also given by C.B. Van Al only. 6 CO2 plus 12 H2O in the presence of sunlight and chlorophyll. The structure of chlorophyll includes a phytol chain. Chlorophyll includes a phytol chain, okay, which has magnesium at the center, sorry, a porphyrin ring which has magnesium at the center and a phytol chain. Do remember ma, it has porphyrin ring and phytol chain, chlorophyll molecule. Then this gives out the end product C6H12O6 plus 6H2O and 6 oxygen molecules are liberated out as byproducts. Now most of the times we get questions like match the following or statement based from the early experiments of this chapter. Okay. Carbon dioxide is essential for photosynthesis is said by mole, by moles half leaf experiment, by moles half leaf experiment. Okay. Water, sunlight are essential for photosynthesis is said by Jan Ingenhouse. It is said by whom? Jan Ingenhouse. Okay. Glucose is formed as a product of photosynthesis is said by von Sachs. Glucose is formed as a product of photosynthesis is said by von Sachs. Okay. Oxygen, with respect to oxygen you will get few questions like this. Oxygen is discovered by Joseph Priestley. Okay. Oxygen is liberated out. Oxygen is liberated out at the end of photosynthesis. At the end of photosynthesis, oxygen is liberated out is said by whom? Jan Ingenhouse. Jan Ingenhouse. Okay. And oxygen is coming from water, not from carbon dioxide. It is from water which is the electron donor, not from carbon dioxide is said by whom? C.B. Van Til. It is said by C.B. Van Til. Okay. And action spectra, absorption spectra. Action spectra which tells us about rate of photosynthesis and it shows the only two peaks which is given by only one pigment, that is chlorophyll A, which is conducting maximum amount of photosynthesis by absorbing maximum wavelength of light, that is action spectra. Absorption spectra which shows different pigments absorbing different wavelengths of light. So action spectra and absorption spectra are given by whom? C.B. Van Til. Both are given by both are given by E.W. Engelmann. Clear? So this is what you have to remember with respect to your exam. Every point is important. Carbon dioxide essential for photosynthesis said by moles half leaf experiment. Water, sunlight essential said by Jan Ingenhouse, and he said that oxygen is released as byproduct from photosynthesis. Glucose formed as end product said by von Sachs. Action spectra, absorption spectra is given by E.W. Engelmann. Oxygen discovery is by Joseph Priestley. Oxygen is coming from water is said by C.B. Van Til. Okay. Take down a screenshot if you want, and this is very important. Okay. Yes. Next, if you look into chloroplast structure. What is it beta? Chloroplast structure, wherein chloroplast is double membrane bound, where you have outer membrane and inner membrane. Chloroplast is double membrane bound, where you have outer membrane and inner membrane. Inside chloroplast you will have sac-like structures, called as thylakoids, sac-like structures called as thylakoids. Okay. Stalk of sac-like structures called as what ma? Grana. Okay. Thylakoids are membranous structures and the membrane of thylakoid is called as thylakoid lamellae. Thylakoid lamellae. Okay. Two stalks are connected by, that is two grana are connected by stroma lamellae. Two grana are connected by what ma? Stroma lamellae. And as we have learnt in the cell, the unit of life, wherein chloroplast is what organelle ma? Chloroplast is a semi-autonomous or self-replicating organelle. It is also called as semi-autonomous or self-replicating organelles. It is because it has a double-stranded circular DNA inside it. Chloroplast has double-stranded circular DNA which synthesizes the world's abundant protein, that is rubisco. It synthesizes world's abundant protein, that is rubisco. And the fluid filled space inside the chloroplast is stroma. Stroma is the fluid filled space inside the chloroplast. And inside chloroplast you will find what type of ribosomes? 70S ribosomes. Inside chloroplast you will find 70S ribosomes. So yes, if you look into the chloroplast, it is a double membrane bound organelles. The membranous sac-like structures are called as thylakoids. The stalk of sac-like structures called as grana. One grana is connected to other grana by what ma? Stroma lamellae. Stroma lamellae, and the fluid filled space inside it is stroma. What type of ribosomes are present? 70S ribosomes. As chloroplast is having double stranded circular DNA, it is called as semi-autonomous or self-replicating organelles. Now if you look into photosynthesis, okay, photosynthesis, wherein two reactions occur inside the chloroplast. Okay, photosynthesis is conducted in two steps. Okay, the very first step is light dependent reactions, which is also called as light reactions, as they are directly dependent on light. Second are dark reactions. Calling light independent reactions as dark reactions is a misnomer, because this light independent reactions are not taking place in the absence of light, but they are dependent on the products of light reaction. Dark reaction or light independent reactions, which is also called as biosynthetic reactions. Biosynthetic reactions, these are dependent on the products of light reactions. So light reactions are done in three steps, 1, 2 and 3, 3 steps. The very first step is absorption of light in the form of photons. Absorption of light in the form of photons. Second is photolysis of water. Second is photolysis of water and release of oxygen. Photolysis of water and release of oxygen. Third is formation of assimilatory power. Formation of assimilatory power, which is both ATP and NADPH. Both ATP and NADPH. Utilizing this assimilatory power, carbon dioxide is reduced to glucose. Utilizing this assimilatory power, what happens ma? Carbon dioxide is reduced to glucose in stroma. This is conducted through many cycles, be it C3, C4 or CAM cycles in different plants. Now if we look into the location, okay, absorption of light in the form of photons takes place in thylakoid lamellae. Thylakoid lamellae or thylakoid membrane, wherein it has photosystems. It has what ma? Photosystems are present in the thylakoid lamellae. Photolysis of water takes place in thylakoid lumen or towards the thylakoid lumen in the inner membrane, okay, inner membrane of thylakoid lumen, inner membrane of thylakoid lumen. Yeah. Next, formation of assimilatory power, both ATP and NADPH. This is formed in stroma. This is formed where beta? In stroma. So, all the sites of the reaction where it is happening is very much important for you. If you want, you can take a screenshot of it. Next, yeah, as we have discussed, thylakoid membrane, where the first step of light reaction, absorption of light in the form of photons takes place. If you consider this as thylakoid membrane, okay, wherein it has photosystems, right. So, yes, this photosystems, two photosystems are present. Two photosystems are named after their discovery. First, photosystem 1 is discovered and then photosystem 2 is discovered. But when you look into the placement in the thylakoid membrane, photosystem 2 is arranged priorly to photosystem 1. And here the maximum absorption of photosystem 2 is 680. Okay, it is what ma? 680. 680 nanometers. Here it is 700 nanometers. Okay. So, yes, in this photosystem, initially you will be having light harvesting complex. Light harvesting complex, which is also called as antennae or accessory pigments. Accessory pigments, wherein different pigments here in the photosystem as light harvesting complex, antennae pigments or accessory pigments, which include carotenoid xanthophylls mostly, will absorb light at lower wavelengths. Okay, at different wavelengths of light, they will absorb the light in the form of photons, and here then they will release out the electrons. Okay, here inside the lumen of the thylakoid, if this is the lumen of the thylakoid, this is the lumen of the thylakoid. Inside the lumen towards its inner membrane, inside the lumen towards its inner membrane, water molecules are present. Okay, here accessory pigments or light harvesting complexes, whichever are there, absorbing light in the form of photons. Okay, here they will release out or they will break this water molecule, absorbing light in the form of photons. They will break the water molecule into 2 electrons plus 2 H plus oxygen atom, half O2. Okay, this is important. One water molecule is photolysed into 2 electrons, H plus and half O2. 2 electrons whichever are there, they get excited and the energy is submitted to the chief pigment, that is chlorophyll A, which is called as reaction center, which is called as reaction center. So, yes, in the photosystem which is present in the thylakoid lamellae, the photosystem which is present in the thylakoid lamellae, accessory pigments or antenna pigments, light harvesting complex is present. They will absorb light at different wavelengths. The photons whichever are absorbed will photolyze the water molecule which is attached to the inner membrane of the thylakoid into 2 electrons, 2 H plus oxygen atom. These electrons get excited and they are submitted to the reaction center, that is chlorophyll A, which is present in the thylakoid lamellae. Once it is submitted, here you understood 2 steps, no ma? Absorption of light in the form of photons, second is photolysis of water molecule inside the thylakoid lumen. Now, third step, how ATP and NADPH are formed. Here comes the concept of phosphorylation. Here comes the concept of phosphorylation, which means addition of phosphate group to ADP and forming out ATP. Okay, and forming out ATP, and formation of the assimilatory power also you will understand here how it takes place. So, phosphorylation you can see it in two organelles beta, one is chloroplast and mitochondria. In chloroplast the phosphorylation whichever is taking place is under the sunlight. So, it is called as photophosphorylation. This is called as photophosphorylation. This is of two types, cyclic photophosphorylation and non-cyclic photophosphorylation. Cyclic and non-cyclic photophosphorylation. First let us understand non-cyclic photophosphorylation. As we have understand, if this is the thylakoid lumen, okay, and this is the thylakoid lamellae, let us consider. Here is the photosystem 2 and here is the photosystem 1. Clear beta? So, here accessory pigments are there, it has absorbed the light in the form of photons, and water molecule is photolyzed to 2 electrons plus 2H plus half O2, which is the oxygen atom. 2 electrons are excited to the reaction center, that is chlorophyll A. Till here is it clear? Now the Z scheme starts. Here it is uphill, okay. The electrons are travelled in the upstream to the chlorophyll A. Now downhill movement starts, that is chlorophyll A, the electrons are transferred to pheophytin. What is it ma? Pheophytin, which is the primary electron acceptor in the Z scheme. Pheophytin will give electrons to plastoquinone, then to this plastoquinone, once it gets the electron, H plus is coming from stroma, okay, it gets attached to H plus which is from the stroma, and it turns out to be plastohydroquinone. H plus lands to the thylakoid lumen again, and 2 electrons are given to plastocyanin, okay, 2 electrons are given to cytochrome B6 and then to plastocyanin. Just a minute, 2 electrons are further given to, just a second ma. Yeah. So, first chlorophyll A to pheophytin, then pheophytin will give to plastoquinone. Plastoquinone will immediately accept a proton from the stroma and it turns out to be plastohydroquinone, where protons are given to thylakoid lumen and 2 electrons are transferred to cytochrome B6 complex. What complex? Cytochrome B6 complex. This further gives out electron to plastocyanin. This further gives out electrons to what ma? Plastocyanin. From the plastocyanin, 2 electrons are again excited in uphill to chlorophyll A, which is of PS1. And here the reaction center is at 700 nanometers. In PS2 the reaction center is at 680 nanometers, where we will also call it as P680 here and P700 here. Clear? Each and every proton molecule you have to remember. From plastocyanin it is to chlorophyll A again, then to ferredoxin, ferrous sulfur complex, ferredoxin and ferrous sulfur complex. After that it gives out to NAD plus. Final electron acceptor here is what ma? NADP plus. NADP plus is present in the stroma, okay. In the presence of NADP reductase enzyme, which enzyme ma? NADP reductase enzyme, NADP plus is reduced to NADPH plus H plus, okay. Now did you understand how NADPH is formed in the plants? Here in the oxygenic photosynthesis, that what happens in the thylakoid lamellae or thylakoid membrane is photons whichever are accepted by the antenna or light harvesting complex are given out to photolyze the water into 2 electrons, 2H plus and half oxygen, which is said to be oxygen atom. Then these electrons are excited to reaction center, that is chlorophyll A, in an upstream then downstream of electron movement starts, which is electron transport chain, where it gives 2 electrons to pheophytin, then plastoquinone. Plastoquinone immediately getting electrons, it will accept proton from the stroma, turning out to be plastohydroquinone, and this hydrogen is given out to thylakoid lumen. Electrons will be carried to the next electron carrier, that is cytochrome B6F. This gives the electrons to plastocyanin. Again uphill movement, that is chlorophyll A, which is present in the photosystem 1, will accept the electrons, then to ferredoxin ferrous sulfur complex. The ultimate or the final electron acceptor in non-cyclic photophosphorylation is NADP plus, which is present in the stroma. In the presence of NADP reductase enzyme, this is turns out to be NADPH plus H plus. Clear ma? This is non-cyclic photophosphorylation. As you can see, uphill movement, downhill movement of the electrons, again uphill and downhill movement. This is also called as Z scheme. This is also called as what ma? Z scheme. Clear? Now, so non-cyclic photophosphorylation is conducted in oxygenic photosynthesis. Okay, oxygenic photosynthesis, that is in thylakoid lamellae and thylakoid lumen. Okay, thylakoid lamellae and thylakoid lumen, and NADPH is formed where ma? In stroma. Till here is this clear? Now, cyclic photophosphorylation is conducted in stroma lamellae. Stroma lamellae. Non-cyclic photophosphorylation includes 2 photosystems, right? Both photosystem 1 and photosystem 2. Here cyclic photophosphorylation includes only photosystem 1, where photosystem 1 take the photons and it gives to the primary acceptor, that is ferredoxin, then to ferrous sulfur complex, then to cytochrome B6F, plastoquinone, to cytochrome B6F, then again to plastocyanin and PS1. Here in between the electron transport, at one place ATP formation takes place. Utilizing the electrons, ATP formation takes place at one place, that is from ferrous sulfur complex to plastoquinone, or PQ from PQ to cytochrome B6F. So, here the electron transport chain is from photosystem 1 and later it comes to photosystem 1 only. This is cyclic photophosphorylation, where you will see it in anoxygenic photosynthesis or bacterial photosynthesis. Only one photosystem is involved and that is present in stroma lamellae. This is about cyclic and non-cyclic photophosphorylation. Till now you have understood 2 steps. One is absorption of light in the form of photons, second is photolysis of water, and third, from formation of assimilatory power you have understood how NADPH is formed. Now from chemiosmotic hypothesis, which is given by Peter Mitchell. Okay, we will understand how ATP is formed in the stroma. If you clearly look into the concepts, because of photolysis of water in the thylakoid lumen, because of photolysis of water, H plus concentration is high inside the thylakoid lumen. When H plus concentration is high, pH will be low. Low pH in the thylakoid lumen and high concentration is seen. First is because of photolysis of water, and second, during the time of non-cyclic photophosphorylation, the primary electron acceptor is binding with a proton, right, which is present on the outer side of the thylakoid membrane. This plastohydroquinone is again bringing the H plus from the stroma. Whichever H plus is present in the stroma, that H plus is brought out by plastohydroquinone into the lumen. And third step is, whichever minimal H plus is present in the stroma is binded with NADP plus, forming NADPH. So, 3 steps ma, what you have to remember. First is because of photolysis of water in thylakoid lumen. Second is H plus is passed from stroma to thylakoid lumen by plastohydroquinone. Okay? And third is in stroma whichever H plus is present, that is binding with NADP plus and forming out to be NADPH. Because of these 3 activities, H plus concentration in the stroma is very much less when compared to the H plus concentration in the thylakoid lumen, because of which an electrochemical gradient will be generated. What is that ma? Electrochemical gradient, okay, or a proton gradient. Because of this proton gradient between stroma and thylakoid lumen, the stroma has less H plus and thylakoid lumen has more H plus, which is highly concentrated, and pH is low inside the thylakoid lumen. ATP synthase pump which has both F0 and F1 complex. ATP synthase pump has what ma? Both F0 and F1 complex. ATP synthase is a channel which is operated in the thylakoid membrane to pump H plus ions from lumen to the stroma, wherein ATP synthase has F0 channel which is embedded inside the membrane, and F1 will face towards the stroma. At one turn, at one pump, it will release out 4 H plus, which combines ADP and inorganic phosphate which is present in the stroma and forms one ATP molecule. This forms one ATP molecule. Clear? So, this is how the ATP molecule is synthesized, and this is explained by chemiosmotic hypothesis, which will balance the proton gradient. Okay? So, here proton gradient is generated and a pump is activated, that is ATP synthase pump, which has 2 channels. One is F0 complex which is embedded inside the membrane, and the other is F1, which where the where the hydrogen protons are pumped from the lumen to the stroma. At one turn 4 H plus are pumped, which combines ADP and inorganic phosphate, forming up ATP. Clear? This is the chemiosmotic hypothesis. So, here with this we have clearly understood the light reaction, cyclic non-cyclic photophosphorylation and chemiosmotic hypothesis. In coming up class, we will discuss about all the cycles which are involved in dark reactions, and we will also discuss photorespiration and factors affecting photosynthesis. Thank you.